This presentation looks at stimulation of glucagon transcription in human mesenchymal stem cells by adenoviral vector by Veronica Franklin, Nida Manan, Jyotika Bahl. The aim of presentation is to review the original paper, which reported that the viral vector system could impact the differentiation capacity of human mesenchymal stem cells by affecting the chromatin structure, thus leading to initiation of endocrine pancreatide transcription. Human mesenchymal stem cells are multipotent, meaning they can differentiate into a variety of cell types, and the stem cells were collected from surgical waste material in surgeries for this experiment. Adenovirus. An adenovirus is considered as the largest among the non-enveloped viruses. This virus contains a double-stranded DNA. It has been proven that this virus is the cause for about 10% of upper respiratory infections in children and adults. Adenoviruses have a gene region called E1. It is associated with cellular transformation and activating gene expression. Deletion of this region allows a new gene to be inserted. In this way, the adenovirus acts as a vector carrying the recombinant DNA. Region 1 E1 deleted adenoviral vector increases differentiation and promotes glucagon gene expression in human mesenchymal stem cells. The adenoviral vector that was used in this experiment was shown to be most effective compared to other vectors.
Methodology: polymerase chain reaction. PCR is used to exponentially amplify a few copies of a template DNA. Reverse transcriptase can build DNA strands based on an RNA template. Real-time enables both detection and quantification of the targeted DNA molecule. In this experiment, cellular RNA was extracted using Trizol, and approximately 500 nanograms of RNA was reverse transcribed using a superscript RT2 kit. It is then amplified by a real-time PCR interplicate, and this was done to ensure reliability and accuracy. Results obtained in the experiment. This graph shows the relative gene expression of insulin, glucagon, and somatostatin, which inhibits the secretion of other hormones. Human mesenchymal stem cells expressing PDX1, NGM3, and MAFA were infected with the adenoviral vector. The E1 deleted region on the adenovirus carries elongation factor 1 alpha promoter-driven DS-Red reporter gene. In this experiment, they used human adenovirus elongation factor 1 alpha DS-Red F50. Elongation factor 1 alpha promotes transcription, therefore it increases gene expression of glucagon. As the results shows, with the adenovirus present, the gene expression of glucagon increases significantly, whereas it has only minor effects on insulin and somatostatin gene expression.
In order to determine the effect of the adenoviral vector on glucagon gene expression, they compared gene-bearing human adenoviral elongation factor 1 alpha to high-capacity adenoviral FLPE F50 helper-dependent adenoviral vector. This is shown as HD on the graph. No glucagon gene expression was detected on human mesenchymal stem cells expressing PDX1 that were exposed to high-capacity adenoviral FLPE F50 vector. However, transduction with first-generation adenoviral vectors increased glucagon gene expression by approximately 20 times. This suggests that glucagon expression in PDX1 human mesenchymal stem cells can be attributed to the presence of adenovirus protein-coding genes. Discussion of the experiment. The human mesenchymal stem cells used in this experiment were derived from the bone marrow that was collected during surgeries. Mesenchymal stem cells are easy to isolate, purify, and expand. Therefore, they are considered safe. They are also used in a large number of clinical studies. Combined effect of PDX1 expression and adenoviral vector transduction leads to the initiation of stem cell endocrine differentiation. The adenoviral vector affects the chromatin structure of mesenchymal stem cells by making specific genes more accessible to transcription factors. In conclusion, first-generation adenoviral vectors can have profound effects on the differentiation capacity of human mesenchymal stem cells. Such vectors have been successfully used for differentiating liver, pancreatic duct, or exocrine tissue cells by overexpression of key transcription factors. Directed differentiation experiments that rely on first-generation adenoviral vectors should be interpreted cautiously, as these gene carriers can contribute to the modulation of the cellular differentiation pathways under investigation.
Thank you for listening.